Hello everyone, today I will be talking about CLDN1 and its role in HCV entry into cells. I'm going to begin with this review article I found. I'll try to keep it quick just because I know that Sujana and Lawrence probably already covered some of this information, I would guess. But it begins with the structure of HCV within the blood. I thought this stuff was pretty interesting. It details how in the bloodstream the envelope of HCV associates with certain apolipoproteins that help it associate with hepatocytes. For example, ApoE and ApoC1 assist in the association of the HCV particle to heparin sulfate proteoglycans, or HSPGs, while ApoE and ApoB help the HCV associate with low-density lipid regions. I assume that all of this is just to help it get in close contact with the hepatocytes to facilitate the first true HCV receptor binding, which is the SRBI protein on the hepatocytes. Essentially, when the HCV gets close, the E2 glycoprotein binds to this SRBI protein and triggers the whole, entry process. Essentially, once it binds, the apolipoproteins on the envelope of the HCV are removed, which reveals another binding site on the E2 glycoprotein for CLDN1. That binds to the E2, and after that, another protein is recruited, which is the EGFR, or the, epidermal growth factor receptor. Altogether, this collection of proteins is called the HSCE complex. And once formed, it begins to move, slowly move towards the tight junctions where it associates with the CLDN1 and occludins. After that, it undergoes a clathrin-mediated endocytosis, and here are some good questions that I thought the review paper brought up. I think number one, there's the question of, does HCV preferentially bind to claudins that are within the tight junction, or is it claudins that are free-floating in just the basolateral membrane? There's evidence to support both sides. I will cover it in the next slide. But maybe I thought a way that you might be able to, test this is maybe you could create a cell line with a mutation in the EL1 or extracellular loop of CLDN1 that could allow for an inducible interruption of the transconfiguration barrier that formed the tight junction, the claudin tight junction barrier. From there, maybe you could track the uptake of HCV via HCV-PP. The second question that I was kind of curious about in the paper was they were talking about how it was unclear if the HSCE complex caused a conformational change in the HCV that allowed it to interact with the claudin, or maybe the HSCE complex just kind of helped it move towards the tight junction, and HCV, just the native HCV, can bind to the claudin. I thought that was just kind of interesting, and I think that it could use some more investigation. Back to the first question. Basically, the paper was wondering if the HCV would bind to claudins that were bound to a tight junction, or that were not bound to a tight junction. Here's some evidence to support both. So there was an experiment done where they had a cell expressing the wild-type CLDN1 protein. Everything was the same, except that when growing the cells, they separated them to, prevent the formation of tight junctions. And what they saw was that they saw a decreased uptake in HCV. This supports the theory that HCV binds to the tight junction associated claudins because there were no tight junctions and there was a decrease in HCV intake. Furthermore, there's another paper that I'm going to talk about that was talking about MA026, which is an anti-HCV compound. what they found was that when hepatocytes were subjected to this compound, there was a complete loss in tight junction barrier function following binding to CLDN1, right? On the other hand, there is some evidence to support that HCV might bind to just claudins that are not in a tight junction and are kind of just floating in the basolateral membrane. Number one, the HSCE complex, they talked about how it just kind of seemed to diffuse laterally following association with CLDN1. This supports the theory that maybe it associates with the tight junction free claudins. Furthermore, another paper discussed how increased CLDN1 association to tight junctions led to lower susceptibility in HCV. This would support the argument that yes, like if there's more of the claudins in the tight junction barrier, that it actually will prevent HCV from entering the cell. Okay, next slide, I'm going to talk a little bit more about the MA026 paper. You can see there on the right, it's a graph measuring different concentrations of this MA026. It's like hepatocytes exposed to different concentrations, and how they uptake, or I guess the rate at which they uptook this fluorescent reporter molecule. You can see at the highest concentration at 3 micromolars of MA026, you see the unrestricted, I guess uptake of this fluorescent reporter, as seen by the steady increase in concentration, while at the lower concentrations you see the tight junction barrier is maintained, and none of this fluorescent molecule is being imported into the cell. I thought that this paper was just kind of interesting to look at, because even though it's not entirely clear if the anti-HCV properties are due to the loss of the tight junction barrier function, I suspect that it could be also that this molecule is just binding to the CLDN1 itself, and maybe it's like competing for the binding site. I thought it's just something interesting to look at, and perhaps yes, the loss in the barrier function doesn't, or I guess prevents HCV from associating with claudins and entering into the cell. That's all I got for tonight, and thank you. Oh wait, I have another slide. Okay. This one is talking about the same, I have a whole nother experiment. I forgot about this one. So this one's talking about how, yes, how claudin-1 and occludin proteins are down-regulated after HCV infection. I thought this one was just kind of neat and interesting. I didn't know that, I didn't expect this result, but it basically talked about how there's an unexplained pathway as of right now where the expression or the intake of HCV, just like it says, decreases the expression of claudin-1 and occludin. They suspect that it potentially could be to decrease the chance of superinfection, or the chance of more HCV molecules infecting the cell following an HCV infection. In figure A, you can see that when the cell was exposed to HCV-CC, you see a decrease in the claudin-1 and the occludin via Western blot. And then on the right, they wanted to really pinpoint which protein it was that was causing this decrease in expression. On the left, you have the FL core, which is just all the main, it's like the E1, E2 dimer, as well as the, I forget exactly what it was. I think it was just the core protein. With all of that together, you see the decrease in claudin. However, when they added like a mutated core, which had a truncated amino acid structure, you see no loss in the claudin, suggesting that it is some unexplained pathway with this core that is causing it. Furthermore, with when they solely express E1, E2, you see slight decreases in claudin 1. However, the E1, E2 dimer, you see a total increase, or total decrease, or total loss of claudin 1 expression. I don't know, I just thought this paper was also kind of interesting. I think it's worth investigating, I guess. Okay, I think that's it. Thank you very much. I'll see you guys later today.